Hey guys, this is Missy. So today we're going to talk about cellular energetics, and really this is just talking about metabolism. So by the end of this presentation, you should be able to answer the questions: how do living systems adhere to the constraints of the universe? And how can a living system maintain order in a universe of increasing entropy? Alright, so let's get started. So we're going to talk about the theory of thermodynamics, and it sounds scary, but it's really not going to be as much information that we need to know as, say, in chemistry. So we're going to talk about just the first and second laws of thermodynamics. So first of all, you guys need to understand matter and energy cannot be created or destroyed, but it can be transformed into something else. So we never lose energy, we never create it. So anytime you have organisms that are doing anything, any kind of work, even if it's just them sitting there, they're always processing energy. And organisms get energy from the sun, they get energy from what they eat, they get energy from what they take in. We can define work, let me go back to that slide. We can define work as anything that requires atoms to be moved around. So cellular respiration, photosynthesis, any kind of metabolic pathway is going to require work be done. Okay, so there's two different types of energy you need to definitely know: kinetic and potential energy. And so you hopefully have seen the words kinetic and potential energy before. Basically, what's going to happen is with potential energy, you're going to have organisms such as these swimmers right here. And this person right here is going to be using or storing potential energy until they dive off, becoming this person who's now going to then be releasing kinetic energy. So the more potential energy we have, we can say the more organized, the more stable a system is. So as you can see in these pictures, we've got the diver who is on the top of the platform. We've got molecules that are pretty nicely packed together, and we've got molecules that are all bonded together. As you release that potential energy and transform it into kinetic energy, we see the diver has jumped off. We see those atoms are now all around. Our molecules are spread all around. And then we see these molecules spread around as well. Okay, so let's talk really quickly about Gibbs free energy. So basically what Gibbs free energy is talking about endogonic reactions and exergonic reactions. So we do use this nifty little equation, and basically what you see here is that delta G is the free energy, delta H is the enthalpy, so the energy stored in a substance, T is temperature, and then S, delta S, is the entropy. So make sure you get this equation set straight in your mind, because you can be given any of these variables and solve for any of them at any time. One thing you want to make sure you pay attention to is in Gibbs free energy, it's going to be used to determine if a process can happen spontaneously or non-spontaneously. And I'll get a little bit more into that in a moment. Okay, so let's talk about exergonic reactions. So when you have an exergonic reaction, it's releasing energy. So the matter is converted from a higher energy arrangements to lower energy arrangements. It happens spontaneously once it's initiated, and then once it's initiated, it kind of continues until completion. This change in free energy, so the delta G, sorry, that looks terrible, is going to always be a negative number. Okay? So over here you've got reactants where they have more free energy, so energy actually gets released, and then the products will have a lower energy, free energy, than the reactants did once the reaction goes to completion. Again, remembering the delta G here is going to be a negative number. Okay, if you have an endergonic reaction, what's going to happen is it requires energy to occur, and so you're converting matter that has lower energy to higher energy. This cannot occur spontaneously. So in all intents and purposes, it's going to be a free energy that's positive. So the delta G is going to be a positive number. Okay, so over here it shows you the reactants are lower energy. You need to input energy into this process. The products come out with a higher energy. So your delta G, the energy that's required to run the reaction, is going to be more than 0. So it'll be a positive number. So typically biological systems use exergonic reactions in order to supply the energy necessary to run endergonic reactions. So in this example right here, we've got more free energy, so a higher delta G, so it's less stable, but it gives you great work capacity. When you have a spontaneous change, the free energy of the system decreases, so now you become less than 0, and then the system becomes more stable, and the released free energy can do work. It can be used for something else. So then you go back to a reaction that needs that energy. Something that has less free energy is more stable, less work capacity, which is why we need this middle section here to create that energy that then a less energy system is going to use. Okay, obviously living systems aren't the only system that exists that needs energy. There are tons of examples in physics and chemistry. In this picture here you see a dam. The energy that it creates by just harnessing that hydropower is going to make electricity. And then obviously harnessing the power of the sun, it's a ton of energy, and you're really not going to get a whole lot of realistic applications for how much energy that sun produces. So the second law of thermodynamics says that any closed system will tend toward a state of maximum entropy. So this is true for the whole universe. Portions of the universe can function as an open system, but usually we're going to see systems that are mostly on the closed end. But again, it just depends on how you define your system. You can use energy to decrease an open system's entropy. So let's see some examples. So we already know that life is highly ordered. We talked about that in chapter 1. Organisms use the energy they have to power cellular and organismal processes that decrease their entropy. This process increases the entropy of their surroundings. So they themselves have less entropy, but then that process has made the surrounding area have an increase in entropy. We all know life requires energy input. In order for a living organism to stay alive, they need nutrition, and that nutrition provides the energy that they need. Okay, so let's talk about what an open versus a closed system is. If you have closed systems, they are going to tend to be free of free energy. There's an absence of free energy, and they reach a state of equilibrium between its inputs and outputs. So in this example, in the picture, we've got a little system here where the water is hydro flowing through these two compartments, but it'll begin to stop once there's an equilibrium reached, and that means that there's no change in free energy. Okay, so when you have an open system, you have an ability to not reach equilibrium as long as you're receiving inputs of energy and you're able to produce the outputs. Okay, so there's no limit to how complex an open system is, as long as there's enough energy for that system. So if we see here in the image over here, you've got the same pulley system, but now there's an ability to keep funneling that water until then we actually have an output that goes somewhere. It doesn't get destroyed, but it goes somewhere. When you look at life as a whole, it's typically always an open system, and it's usually more interesting. Okay, here's a closer look at those pictures. Equilibrium typically equals death. So it is not ideal for a living system to reach equilibrium. There's always going to be a need for an influx. Okay, so there's two things we need to talk about here. The cellular energy theory, so now we're going to apply all of what we just learned about to two things that have to do with the cell. One of them being ATP. Okay, it's short for adenosine triphosphate. It's a short-term energy storage and it releases energy in cells. So cells can't use carbohydrates and lipids in its raw form, so there's a bunch of metabolic processes that will convert that energy into ATP. Okay, so we talked about macromolecules, and as you guys can see, there is a bond between a phosphate group and then a nucleotide right here. Okay, because of that triphosphate, the nucleoside triphosphate, it's going to make it unstable, which actually means it's really great as a renewable energy source for the cell. A lot of free energy is released when the bonds are broken, which is enough energy that the cell can then harness and use. Uh, and that then, as you can see, gives you the energy, but also gives you a new molecule called ADP, and that molecule can then be reused in the cycle when a new phosphate gets added. Okay. Much of the work done by cellular proteins is mediated by the addition and removal of the phosphate groups from ATP, and it's removed by proteins to other proteins. These are called kinases and phosphatases. Okay, we'll talk more about those when we get to communication between cells, but it's important to remember that kinases and phosphatases are used a lot in the facilitation of removing that phosphate group to create this energy. Okay, two examples here. You've got transport work, so cells transporting molecules through active transport. That energy is needed in order to facilitate that protein to open. In the mechanical work, you've got ATP binding to a non-covalent motor protein, and then once that ATP binds, it's hydrolyzed, and now that protein. Okay, so let's talk about metabolism. So a lot of time in metabolism, people think of it in terms of, oh, I'm breaking down my food, and that's how fast I can break it down and not gain weight. Well, it's a little bit more complicated than that. So one of the things to consider when it comes to metabolism is that you need energy to have a process occur, and then you get energy that is released as a result of that. So let me see if I can get my drawing here. All right. So what we're going to have here is energy from catabolism, and then we're going to have energy from that's released, and that's the cellular work that's going to be endergonic, so it's energy consuming. So energy from catabolic reactions, such as cellular respiration, are used to power the synthesis of ATP from ADP. Okay, and that's where those phosphate groups come into play. Then when you have ATP using anabolic reactions, those are the ones that are going to require there be chemical energy. And I apologize, I totally messed that up. So before we end this video, I'm going to talk a little bit about kinetics. So the reason why we need to know about kinetics is because of its activation energy application, and we're going to apply that to when we talk about enzymes. So if you look at this diagram here, let's see if I can get this pen to work correctly. You're going to have your reactants, and they're going to require some sort of activation energy. That activation energy is that place where it goes to eventually overcome that hump is called a transition state, and then we get a decrease in activation energy until we get our products. In this case, our delta G will be less than 0, which is what we want. So why is that important? Well, because we can use a catalyst. A catalyst is any substance that can increase the rate of a chemical reaction while not actually being in the reaction itself. What's important here, it lowers activation energy. Okay? And because they don't participate, they are reusable. So if you see here, that same reaction, we've lowered the activation energy, so it takes significantly less energy to achieve the same results. So after watching this video, and hopefully I didn't bore you to tears, You should make sure you can explain how living systems adhere to the first and second laws of thermodynamics. You should be able to explain how living systems can increase in order even though the universe is moving toward a state of maximum entropy, which again, entropy just means chaos. Compare endergonic and exergonic reactions. Compare open and closed systems. You should be able to explain how ATP allows for cellular work and explain the effect of a catalyst on a reaction profile.